This voiceover talks about regulating skeletal muscle contraction, and as you'll see, the regulation is by calcium ions. This is a cartoon from another textbook, but somewhat similar to what's in your textbook. Let's anchor it to an EM picture, a longitudinal section from your textbook, showing what the myofibrils look like, with the sarcoplasmic reticulum and T-tubules showing as well. The cartoon now shows a motor neuron, a neuron that innervates a muscle cell, attaching to this muscle at a neuromuscular junction. And we're going to see an animation of the sort of thing that happens. And you will remember that a nerve impulse will be moving down this axon to the nerve terminus, to the neuromuscular junction, at which point some changes will occur that leads to an action potential. So here's our nerve impulse. The membrane depolarizes. Here we have the depolarization and the traveling the propagation of that depolarization along the muscle membrane. Let's look at that once more. There's the depolarization or propagation of that point of depolarization along the muscle membrane until it reaches a transverse tubule where the propagation is then continued down the tubule towards the sarcoplasmic reticulum shown in yellow here. The propagation of that depolarization continues into the sarcoplasmic reticulum, and in the sarcoplasmic reticulum there is an opening of calcium channels and a release of calcium which now bathes the contractile proteins, the actin and the myosin. The ion channels that open up will later serve to recapture the calcium after contraction is over. Well, let's take a closer look at the actin filaments in muscle. Actin is actually associated with many different kinds of proteins that enable it to function appropriately. So here we see some. There are the actins, this twin helix of polymerized globular proteins, actin proteins, and lying along the length about every 7 or 8 actins in this double helix, if you will, are molecules of tropomyosin shown in darker blue. And attached to each of those at about every 7th or 8th actin are a series of small proteins called troponins. These are going to function in the response of actin to calcium. So in this image, we are looking at a cross-section through an actin filament, and the two white balls are the two globular or G actins that are part of a chain, but we've sliced through the actin filament, and so we can only see two of them, one's behind the other. And this is in the region where tropomyosin and the 3 troponins come together. If you were to do serial sections through a thin filament extracted from muscle, you would see this image roughly every 7 or 8 actin monomers. We have names for the troponins. They're troponin T, I, and C. We are not going to be too concerned with what each of them is called or what they specifically do. I want you to get the general picture of how calcium interacts with the troponins, changing the shape of the troponins and eventually the shape of the tropomyosin, and giving access to the myosin heads. So this is that cross-section. In the presence of calcium, as you can see, the troponins and the tropomyosin have shifted position, exposing 2 sites, one on each actin in this monomer, to which the myosin heads can now bind. And for as long as there is calcium in the vicinity of the myofibrils, able to interact with the actin, myosin heads will now be able to go through their conformational changes and draw actin filaments along myosin. It's all a matter of shape, remember? Conformational changes induced by calcium binding to troponin C, which causes a conformational change in the other troponins and in tropomyosin, and that conformational change is reflected as a movement away from the sites where myosin can bind. Let me add that during muscle contraction, you are sending nerve impulses to that neuromuscular junction and continually firing off action potentials which get propagated down the T-tubules to the sarcoplasmic reticulum, and calcium continues to bathe those myofibrils. When you no longer wish to contract that skeletal muscle, you stop sending the nerve impulses, and the calcium is retrieved back into the sarcoplasmic reticulum by a calcium pump, and the calcium no longer bathes the actin. The tropomyosin and the troponins resume their position, occluding or covering the myosin binding sites, and contraction basically stops and the muscle relaxes, or can be relaxed. A few notes on how these activities in the sarcomeres and in the myofibrils actually generate force. In order to do that, the actin filaments must be pretty firmly attached to the Z lines, and then ultimately, in order to pull the membranes of a muscle, the actin filaments, the sarcomeres, have to somehow be attached to membranes. So let's take a look at this. These are immunofluorescence stained cross-sections through a Z line. And there are two proteins shown here. The immunofluorescence is due to fluorescing antibodies against a protein called alpha-actinin and another one called desmin. This is a bunch of Z discs in register, that is aligned at the same position along a muscle. And so each bright circle in the alpha-actinin stain represents one Z disc, and you can see all the Z discs are held in register. They're all next to each other. They're not staggered in the myofibrils. And if you look at the desmin stain, you see much the same thing, except the desmin stains the circumference of the Z disc, and alpha-actinin seems to permeate the Z disc. Well, that has some consequences for our understanding of what's going on here. Let's draw a cartoon of Z discs. Here we have 7 Z discs out of the many that we just saw. And in the middle of those Z discs, those very tiny white dots are meant to illustrate alpha-actinin linking actin to the Z discs. So alpha-actinin is an actin-binding protein that attaches the ends of actin to the Z discs. And the red stuff, that's the desmin that actually holds the Z discs together. So the Z discs, being firmly attached to actin, will be pulled during contraction towards one another along the length of a myofibril. And they will all be pulled at the same time because desmin is holding all of them in register. That is, all of them at any given point in a myofibril are being held together in register. And that brings us to the end of this presentation.